video participants. Can you see that on yours, or is it just on mine?
Yeah, you're viewing your screen. There's a caption live on custom live stream service.
Okay.
I think it's on yours probably.
It's just on mine. Okay. Okay.
So the participants, that Zoom can be either in the bottom of the screen or on the top.
Okay. Yes. Shall we make a start?
Sure. Let me give a brief introduction. So hello and welcome to the 6th Champions Talk. Today we are very fortunate to have with us Professor Bestra. Professor Bestra is the current president of the Microbiology Society UK. He is Borderic Professor in Microbial Physiology and Metabolism in the School of Biosciences at the University of Birmingham. Professor Bestra heads a world-leading multidisciplinary team investigating key aspects of the microbial physiology of the mycobacterium tuberculosis cell wall. He has been awarded and successfully managed over 50 research grants valued at over £35 million from the Wellcome Trust, the Biotechnology and Biological Sciences Research Council, and Medical Research Council. He has published over 480 internationally peer-reviewed articles, including in Science, Nature, Immunity, Journal of Experimental Medicine, and Proceedings of the National Academy of the Sciences, USA. He also published reviews, book chapters, and been more than 34,000 times as an H index of 106 at the Google Scholar. It is indeed a pleasure to welcome Professor Bestra. Today, Professor Bestra will share his academic journey as a TB researcher. We hope that this talk will inspire many young audience here to remain motivated in their career path as a researcher. With that, we welcome in Professor Bestra to give a talk.
Thank you, Dr. Mitra. So what I'd like to do is talk a little bit about my sort of journey as an academic. So I think I go right back to the beginning, and I'll share some sort of personal insights into where I come from. So my parents moved to the UK from India in 1952. So I'm really the first generation migrant to the UK. And so I was born in 1965 in Sorrento Hospital in Moseley in Birmingham, and sort of quite interesting sort of caveat and fact here is that this was one of the first special care units that was set up for premature babies. And in fact, I was actually born premature by several months. A lot of my family still live locally in Birmingham, so we have strong roots here. I attended a local primary school in Sparkbrook, which was the sort of norm in terms of education in the UK. My parents were a bit reluctant. We have this what's called as the 11+ system in the UK, where you can do examinations to go to grammar school. But they were a bit reluctant to do that at the time because it impinged on where you would go in your secondary school if you were not successful in terms of getting into grammar school. So I went to a local comprehensive, also in Sparkbrook at the time. What was interesting that when I sort of finished my A-levels at secondary school, I didn't really do that well. And I actually wanted to do pharmacy. And I think this is a trait that's sort of built into sort of a nation background in terms of, parents always want their children to pursue careers that they see as professions that they can look to and aspire to. So you see a very strong sort of link to people wanting to do sort of medicine, dentistry, law, accountancy, engineering. So at that time, I was thinking of wanting to do pharmacy, but I didn't get in. So this presented some challenges for me in terms of what I would do next. And I was sort of fortunate at the time that within the university system, you had a system where you could have what was called CAP, a continuing application procedure, where if you weren't successful the first time, you could try again and pick another subject. So really, I only knew the basic sciences in terms of A-levels, and those were the sort of degree courses that were familiar to me. And I was probably a little bit naive then in that, after being through the whole university system, there's a lot more courses out there and a lot more choices if you really look hard. But through CAP, I was interviewed at Newcastle University by Dr. David Mencken, and there's a picture here on the right. And so it's quite a sad story in that, so David passed away last year, and he's been quite a sort of mentor to me over the years, and people within the mycobacterium community will know him very well in terms of his what's called the Mencken model of the cell wall. So he interviewed me, and I think he took to me, he thought that my interest in sports, and, I like football, I like to play cricket, and he thought I'd be an outstanding person to have at the university as an undergraduate. And that was the end of the conversation. So I went to Newcastle as an undergraduate student studying chemistry. At the end of my undergraduate degree, I wasn't sure what I was going to do. My family are quite successful business people. Would I actually go back to Birmingham? But he actually persuaded me to do a PhD, and it hadn't even crossed my mind to do a PhD at the time. And he thought, oh, there's something special here, and he could be quite successful in this as a career. So he encouraged me to stay, and I entered into the PhD program in chemistry and organic chemistry at Newcastle with David. After completing my PhD, I had a number of choices at the time. One was to go into industry with either Glaxo Wellcome. I had another offer from International Paints or the Atomic Weapons Establishment, all quite varied sort of positions in industry. Ideally, I wanted to go and work at Glaxo Wellcome, but at that time in the 1990s, we were starting to head into a recession, and a number of the positions in industry were frozen. I didn't really fancy either of the other two, and having a long chat with David, he said, well, why don't you actually go and do a postdoc in the States? So he had a conversation with this guy down here at the bottom, who is Professor Patrick Brennan at Colorado State University. And basically through a few phone calls, I didn't even meet, Pat Brennan before, never spoken to him. He basically sent me an email saying, you can come along, you can join my group as a postdoc. So you can see a lot of this has all happened by chance and serendipity in terms of where I've ended up. So I ended up spending 8 years at Colorado State University. And really, I went from being someone who was interested in organic chemistry and lipid sort of chemistry to actually understanding more about the biology of TB. I became quite interested in molecular biology, immunology, and also structural biology at the time. So I became quite rounded in terms of the different disciplines I was interested in. So obviously, working in the States, it wasn't a sort of long-term view for me in terms of a career, because when I looked at the academics in the US, they have quite a different system to the UK. So I was quite interested to come back to the UK at that time. Plus, I'd already started a young family, and I was thinking about the future for them. So I was offered a number of positions over the years. One was as a deputy director of the TB labs at Public Health Labs in the UK. I was offered a readership at Reading and also at the University of Manchester. But what really changed everything was that in 1998, after a long discussion with colleagues at Newcastle, I basically came home, to Newcastle University, where it all started. And Professor Minikin, Bearer, Harwood, Amachi, and also the Dean of Medicine and the Pro-Vice of Research were very keen that I come back to Newcastle to start a new group on TB research. They offered me a readership with a fantastic startup package. And I came back, I was awarded a Lister Fellowship in 1999 and was promoted to a professorate in 2000. I was only 35 at the time. And this is a picture of Newcastle, the medical school, and we were based here on the 3rd floor. And really the whole idea was, could we actually set up a grouping that would achieve funding from the MRC? The MRC were quite interested at the time in these MRC cooperative groups. So we set up one of these, and obviously we needed a number of individuals who would interact together collectively. So obviously these involved myself, Professor Bearer, Dr. Ian Hancock, and Professor Minikin, while we were all at Newcastle. But after time, things change and things move on. Professor Bearer went to Leicester, Dr. Hancock retired, and Professor Minikin was coming to a retirement age as well. So I then started to think about what I was going to do next. And really, when we start to think about the case for this program, and what the MRC was looking for, was really, they were established to tackle tuberculosis in 1913. It's important because of the rates that we see for the disease. And also, we had a strong collection of individuals interested in bacterial cell walls. And we felt this MAGPIE program would be something that would be of interest to the MRC. So, as a one of the things that gone through the whole of my academic career is this interdisciplinary approach that we used for tuberculosis research. So obviously I have a grounding in a number of areas in terms of biochemistry, organic chemistry, immunology, and molecular genetics. But the other individuals that were involved were interested in organic chemistry, Professor Bearer in clinical microbiology, Dr. Hancock was an outstanding biochemist, and Tony Lamas was interested in immunology. And really, we had a number of collaborators all around the world who would work with us. So, one of the problems that we see with tuberculosis, really, when we think about TB, we look at these global incidence rates in terms of MDR and XDR-TB. We have, what I sometimes refer to as a north-south divide, in that you have the southern hemisphere where you have very low case rates, but you have, quite high case rates in Asia, Africa, and South America. And when you start to map onto this MDR-TB, you can see high case rates in Asia, parts of Africa, and South America. And, the case rates that we have are staggering. We have 8 million new cases each year, 2 million deaths each year associated to tuberculosis. And really issues stem in terms of the therapy. We have what's called DOTS, directly observed short course treatment. You have an initial phase of 4 drugs for 2 months. These consist of rifampicin, isoniazid, ethambutol, and pyrazinamide. You have a continuation phase with 2 drugs for 4 months of rifampicin and isoniazid. So really you have a 6-month course of treatment. If you have MDR-TB, this actually continues on for 2 years, and you have the use of second-line injectable drugs that are brought into the therapy. We don't have any treatments for XDR-TB, which is termed extensively drug-resistant TB. And the third issue we have is with HIV and co-infection. Really, if you think of the endemic population that has a TB infection, you have a 5% risk per lifetime of developing TB. If you then overlap with the HIV endemic population, your actual rate of risk becomes 5% per year of contracting TB. So our strategy at the time was really to try and identify drug targets and look at post-pathogen interactions, use functional genomics to identify candidate genes, and really select genes of interest and perform focused studies, which then were underpinned by our different expertises in terms of disciplines that we all had. So the MRC sort of bought into this program of work, and they funded 5 project grants, all in one go in one session at the board meeting at the time. And we had funding also from the Wellcome Trust and the Lister Institute. So we were off and running in terms of setting up the Magpie Group at Newcastle at the time. So as I sort of alluded to earlier, that I was actually looking at where I was going to go next after spending 1998 to 2002 at Newcastle, was that I saw individuals leave the university, people come into retirement, and also being from Birmingham, I was having a family commitment that I should actually try and get closer to the Midlands and be closer to the family. So I ended up accepting a position in the School of Biosciences, which is where I am today. And what I'd like to do now is share a little bit of sort of research that we've done and some of the things that we've done in terms of establishing the biosynthesis of the cell wall of mycobacterial tuberculosis. So it has an underlying sort of peptide glycan, as we show here. It has this arabinogalactan unit, which is linked through a phosphate to muramic acid. We have this unique disaccharide bridge, which consists of rhamnose and N-acetylglucosamine. We then have this galactan backbone, which is a beta-1,5, beta-1,6 galactofuranose backbone. It's around about sort of 30 residues in size. But then at different positions on the backbone at regular spacings, we then have these arabinan chains. And these are quite distinctive. They're arabinose in the furanose ring form, and they have different linkages in alpha-1,3, alpha-1,5, and beta-1,2. And they're put together in terms of different motifs. And each sort of stub is probably around about 30 to 35 residues. So the whole polymer is around about 100 sugar residues. Attached to the termini of the arabinan segments, we have these long chain mycolic acids. These are beta hydroxy fatty acids, and they range in size from 70 to 90 carbon atoms. And really, this is what lends the sort of inertness in terms of what we perceive as the bacterial cell wall in terms of its hydrophobicity and why drugs can't penetrate through the cell wall. Intercalated within the mycolates, we have a number of sort of species-specific lipids. They can either be inert waxes, such as the thiaceryl dimycosyrosates shown here, or you can have glycolipids, such as diacyltrahaloses or phenolic glycolipids shown here. So you can see that this becomes quite a lipid-rich, waxy mesh on the outside of the cell wall. We also have lipoarabinamanan. This is sort of the equivalent sort of teichoic acids for mycobacteria. We have it anchored in the membrane. We have a mannan backbone, which is branched. The linear segment is alpha-1,6 with short 1,2 branches. Then we have the same arabinan segments as we find in arabinogalactan. The LAM is also capped by different residues. So in mycobacterial tuberculosis, you have what we refer to as mannose-capped LAM. So some of the challenges in terms of looking at the biosynthesis of these molecules, they use lipid-linked sugar donors as substrates. So they involve the use of membrane-bound glycosyltransferases, but notoriously difficult to study. And many of the enzymes are essential in TB. The interest in terms of the cell wall is that many of them are the target of frontline drugs such as isoniazid and ethambutol. So what's our current understanding? So really what we do here is we try to utilize different organisms. So we can switch between using sort of mycobacterium tuberculosis, mycobacterium bovis BCG, mycobacterium smomatus, and even a surrogate such as Corynebacteria glutamica. And we can switch between the different organisms depending on the sorts of assays we want to do, whether we can generate recombinant proteins, because sometimes when you generate recombinant proteins, as you switch organisms, you can get better yields, you can get soluble protein. And also in terms of making mutants, whether we're making deletion or conditional mutants, we can also switch in terms of the different organisms because you can get more of a stable phenotype in certain backgrounds. So the biosynthesis starts by the transfer of N-acetylglucosamine from its sugar nucleotide to a polyprenol phosphate, which is embedded in the membrane. We then have the transfer of ramenose by the wobble gene. We then have the transfer of galactofuranose from GLFT. We have 2 residues added. Then a second enzyme, GLFT2, then sort of polymerizes the whole sort of galactan backbone. We then have a flipase. And the reason that we have the flipase is now we start to use what are these unusual lipid-linked sugar donors. So what I've shown here is DPA, which is basically a decaprenol unit, which is a C50 isoprene with a phosphate linked to arabinose. And what we have is the transfer by different enzymes. Here we show AFTA adds at 3 different positions precisely along the galactan backbone. We then have ethambutol, EMB A and B. These are the targets of ethambutol. And these then sort of go on to polymerize the rest of the arabinan segments. And there's a little bit of debate at the time, which I'll come back to in a minute, in terms of how ethambutol works. We then have AFTB, which forms a final sort of capping in terms of the addition of the terminus arabinose residues. And we then went on to describe AFTC, which introduces the branching of the different chains. So in terms of ethambutol, it was long thought that ethambutol was involved in the bulk of the polymerization of arabinogalactan and also involved in lipoarabinomannan biosynthesis. But what we've shown more recently is that it's very much involved in the terminal steps in terms of arabinogalactan synthesis. And really, this was a key publication that we had in Science, and this was a fantastic collaboration with Lu Zhang and Professor Zi Rao from China. It's one of those things where someone sends you an email and you don't know what to make of it, and then you start a dialogue and suddenly you're in a fully fledged collaboration. You've never met the people before, and it's just been fantastic to work with them. So they purified the M proteins, which I mentioned before, membrane proteins, very difficult to work with. And we developed all the biochemistry in terms of looking at how the arabinose from the DPA donor is transferred to this synthetic artificial substrate. We were able to show that it attaches at the 3 position here. And basically, the other thing that we found was that we always had this concept that MA did one thing and MB did another thing. But actually what we found out was that MA and B, they actually form a dimer together, and it's the MAB complex that results in this transferase activity. So this is sort of shown in the schematic here in terms of basically, if we take our controls in terms of no acceptor or no protein, we get very little activity. As soon as we add in our dimer of MAB, we then have transfer of the arabinose residue. If we add in ethambutol, we see reduction in activity. We also made the M. smegmatis MAB dimer, which is active. Again, if you add in ethambutols, you see an inhibition. And then we did a number of point mutations that we showed were important in terms of activity. Similar sort of thing for MC, which is another M protein, which was shown to be involved in lipoarabinomannan synthesis. Again, we used a synthetic disaccharide. But here, the MC is actually involved in the transfer in a 1,5 fashion. And again, we showed basically with smegmatis and TB, MC, again, as a dimer MC, that we could get activity and inhibition by ethambutol. One thing I haven't mentioned is that when we actually purified these proteins, they were purified from mycobacterium smegmatis. They were always associated with an acyl carrier protein. We have no idea what this acyl carrier protein is doing. We don't know whether it's involved in somehow allowing access of our decaprenyl phosphoarabinose substrate to the complex, but this is an unknown at the moment. So these are some more sort of illustrations of the dimers in terms of both the ethambutol, in terms of where DPA is docked, and also in terms of whether we're looking at MTB or M. smegmatis. But I'll just refer you to this paper because it has some beautiful data in there. I'll talk a little bit about what we've been doing in terms of mycolic acid biosynthesis. So as I mentioned earlier, these are sort of the waxy fatty acids that we find in mycobacterium tuberculosis. They have this long alpha alkyl chain, the beta hydroxy fatty acids, that is short alpha chain, which is around 26 carbons. The mero chain is usually around 50 to 60 carbons, and they have different sort of functional groups. Here I've shown an alpha mycoli, which has cis cyclopropane rings. So what's unusual about mycobacteria in terms of fatty acid biosynthesis? Typically what we tend to have is what's called fatty acid synthase 1, which is found in animals. It's a multifunctional polypeptide, and it does all the necessary activities that you have for fatty acid biosynthesis. Mainly in bacteria and plants, you have what's termed fatty acid synthase 2, and it consists of several distinct interacting proteins, so it's a dissociable complex. Mycobacteria is actually unusual in that it has both FAS1 and FAS2. FAS1 forms de novo synthesis of short-chain fatty acids up to C16 to C24, and FAS2 is what is involved in extending these to sort of fully formed mycolic acids. So over the years, we've sort of pieced together the whole biosynthetic pathway in terms of mycolic acid biosynthesis. So one of the first things we did, we characterized the acyl carrier protein, ACOPM, and FABD in terms of generation of malonyl ACPM, which is the extending unit that we have for fatty acid biosynthesis. We then went on to describe FABH, which takes the malonyl ACP with an acyl-CoA to give this keto-acyl ACPM intermediate, which is the first step of FAS2 biosynthesis. We went on to study Cas-A and Cas-B, which are additional keto-acyl enzymes that are involved in elongation. We worked on the dehydratase enzyme, which basically dehydrates this position here to give this enol intermediate within the cycle. We worked on INHA, which is the target of isoniazid, which reduces the enol to the saturated form, and then you go back into the cycle again. And we worked on many of the tailoring steps in terms of desaturasses, the modification of the mycolates, and also how the two sort of alkyl chains are stitched together through a polyketide synthase, and also how the mycolic acids are actually attached to the cell wall and to trihalose through the antigen 85 complex. So over the years, we've done a lot in terms of the biosynthesis of the cell wall, but then what happened about 10 years ago was that we developed an industrial partnership with GSK, and they had this TB screening box where they'd done high throughput screening of inhibitors and identified a large collection of compounds. And they made these freely available to us, and we developed a strategy where we would develop mutants to these compounds, and then we would do whole genome sequencing to see whether we could identify targets. We had a strategy of using quantitative chemical proteomics, and also we developed a screen based on a sort of hybrid of a phenotypic screen and looking at specific targets. So really, the idea was, can we identify the targets? Can then these be informing sort of hit to lead programs? And also, could we understand better metabolic pathways of targets that we were interested in, such as DPRE1 at the time? Is it okay if I continue, or would you like me to stop? Is it okay? Okay. So I'll just give a couple of examples. So one of the ones that we're first interested in were these compounds here, these indazole sulfonamides, and they were shown to be highly active against TB. And what we did was we generated mutants and we identified SNPs in Cas-A, which obviously goes back to some of our interest in terms of mycolic acid biosynthesis. And we were able to show that basically when you add these compounds in, basically what you can see is a depletion of mycolic acid biosynthesis. This is a thin layer chromatogram where we've labeled cells and then purified out the fatty acid methyl esters and the mycolic acid methyl esters. And you can see quite clearly, as you increase the concentration, we're starting to get a decrease in terms of the synthesis of mycolic acids. These were control experiments where you see a similar phenotype with isoniazid, which is the target of INHA, and TLM, which is also a target of Cas-A, you see a similar phenotype. So our sort of biochemical data was fitting our sort of genetic data in terms of Cas-A being the target in terms of within mycolic acid biosynthesis. To look at this further, you can actually purify out the cell wall mycolates, and you see a much more dramatic phenotype in terms of depletion of mycolate synthesis as we increase the concentration of the drug, and the last two other controls that we used. But to provide further evidence, if you overexpress your target gene, you can titer out the effect of the drug. So we went away and we cloned all the different genes involved in the Fas2 biosynthetic pathway. And when you don't have them with drug, you can see that they're all growing quite happily. So when we add in small amounts of the drug at 1 times the MIC or even higher at the MIC, what you can see now is that all the other enzymes that are not involved as a target do not grow because they're killed off, but only the organisms where we've overexpressed the plasmid which has the target, which is Cas-A, which goes back to our whole genome sequencing data, you can see that the cells are now surviving at the MIC. And in fact, if you go up to 8 times the MIC, they're still surviving. So this sort of verifies that our target is Cas-A for these particular drugs. We also had a nice sort of in vitro biochemical assay, which again, we can show that we can see nice activity in terms of Cas-A. And again, when you add in the drug, you can inhibit the Cas-A activity in vitro. So Catherine in the lab, she went on to solve the crystal structure of Cas-A, and it's a dimer. And also she managed to co-crystallize TLM within the active site. So the last bit of the talk is really about sort of MMPL3 and the role of MMPL3. So it's one of these promiscuous targets in terms of TB, in that there's lots of different scaffolds, which are shown here, that target MMPL3. And these are all identified through SNPs in MMPL3. And at the time, it was shown that MMPL3 was involved in the transport of TMM. So therefore, is important in terms of the final location of mycolic acids in the cell wall. So one of the things that we did was we went back and started to look at some of these compounds in terms of what they were specifically doing. So again, if we label cells and we purify out fatty acid methyl esters, FAMEs, and the different mycolic acid methyl esters with increasing amounts of drug. So what you can see here is that basically the FAMEs are not going down in terms of the synthesis, but the mycolic acids are being depleted. If we take the cell wall bound MAMEs, you can see that it's a very distinct phenotype in terms of you're seeing a very pronounced inhibition. So this particular compound fits with its inhibiting sort of mycolic acid synthesis somehow. Now, if you take this is the inactive enantiomer, so it's obviously dependent on the enantiomeric purity of the compound. Here you don't see any effect. Now, one of the things that sort of struck us was that when we started to look at other MMPL3 inhibitors, we didn't see the same phenotype as we were seeing for this compound, where we're seeing quite a dramatic effect. So this sort of suggested to us at the time there was something else going on. Maybe there could be additional targets for some of these MMPL3 inhibitors. So this is when we moved to the use of quantitative chemical proteomics. Basically, what this requires is that you take your hit compound and you basically attach it to a bead. You expose it to the proteome and you look for the capture of its ligand to the compound. And then you remove the ligand through a release procedure, and then you do proteolytic digestion and you look at the fragments. So you're looking at specific binding from a cellular lysate to your target molecule. When we actually did this, we actually identified this particular hit, ECHA6. So we went back and we made a conditional depletion mutant of ECHA6 in Mycobacterium bovis. And what you can see is that when we start to deplete the anhydrotetracycline that we have, you can start to see cell lysis of growth. Again, you can see inhibition of the fatty acid methyl esters here in terms of the MAMEs. So basically, we've established a sort of chemical and genetic inhibition of MAMEs with these experiments. What we did then was, if you do the same experiments like we did with Cas-A, if you overexpress ECHA6, what we can find is that we can actually change the resistance profile. The MICs were quite low, 0.32 micromolar, but when you overexpress ECHA6, the MICs shift to 2 micromolar, an 8-fold shift. Now, you also see in the same sort of protection here, in that when you add in ECHA6 on a plasmid, you don't get the level of inhibition as we were seeing before in terms of mycolate synthesis. So ECH6 is involved in binding long-chain CoAs, and we were able to establish crystal structures where we showed that you can actually bind in a long-chain fatty acyl CoA and also the molecule itself. So we were establishing a sort of picture in terms of, these molecules are targeting mycolic acid biosynthesis somehow, the binding long-chain CoAs. And we were able to confirm some of this through sort of tryptophan binding of different acyl chain CoAs. Clearly, long-chain CoAs were better. And we could also see inhibition when we were adding drug of binding. So we came up with a hypothesis at the time, because ECHA6 is involved in sort of the catabolic pathway in terms of fatty acid biosynthesis. So what we thought at the time was that ECHA6 could be a mechanism by which it provides substrates to Fas2. And this is why when you inhibit ECHA6, we see a depletion of fatty acid biosynthesis. Now, if you go back to MMPL3, why would this be different for MMPL3? Because MMPL3 is involved in a later stage, whereas you wouldn't see the inhibition of Fas2, you just see the accumulation of trihalose monomycolate. So through the chemical proteomics strategy, we were able to dissect basically a new sort of pathway in terms of fatty acid biosynthesis that's involving the recycling of acyl chain CoAs into Fas2 biosynthesis, and was very distinct from studies on MMPL3. A little bit further on, we went on to show through 2 hybrid experiments that basically you could see that ECHA and Cas-A were interacting. So this was then interesting in that this was the key point of the way fatty acids would come into the cycle in terms of Fas2. It's interesting that it also interacts with INHA, which we don't really understand why it would be doing that, but it could be that there could be 2 mechanisms by which different substrates could be coming into the pathway. So I think, really, when I look back at my career, it's really been one of pure chance. Some things have sort of fallen my way, in terms of doing organic chemistry at Newcastle University, when probably I wasn't thinking of that originally. Meeting, Professor David Minikin, who was a key mentor for me in terms of my early career, persuaded me to do a PhD, then persuaded me to go to the US to do a postdoc with Professor Patrick Brennan, staying in the States, then having that sort of, coming back to the UK. And actually, I think some of the things that I found are, a lot of things are by chance. And I think you have to take that sort of gut instinct in terms of where you feel comfortable and really, move when you think it's the right time to move. Colleagues are hugely important. I think collaborations are key to science. I think over the years, I've collaborated with many, many individuals all around the world. And I think that's provides a very fruitful platform in terms of success as well. And also colleagues that, who are supportive of your career. And, a lot of the awards that I've had over the years wouldn't be without the support of colleagues who recognize some of the work that you do. So I think those interactions which you have, with colleagues are very, very important. And I'm always grateful for the support that they provide me. And finally, I'll finish with, acknowledging, I don't do much of the work. It's always people in the lab who do this, and also the different funding sources that we have that support the work. Probably gone over quite a bit, but I'm happy to take questions. So I'll stop the share if I can.
Thank you very much for the talk, Professor Besra. Now the floor is open, and the participants can either unmute themselves or they can write their question in the chat as well. Yeah, Andy, go ahead.
Well, first, thank you very much, Gurjit, for the nice talk. I'm calling from Newcastle University, so I really like that part, especially. So you mentioned that being part of a large collaborative group was really important for your research. So I was wondering, would you mind elaborating a little bit more on how this big group came to be and what your experience was of working with so many collaborators in terms of, I guess, applying for grants, writing papers? How is it working in such a big group?
Okay. Well, as I mentioned, a lot of things are by chance and by taking that sort of gut instinct in terms of where you go. I think in terms of establishing that group of individuals, I think a lot of it came through knowing the individuals in the field that you're working in. So obviously with someone like David Minikin, he was connected with, Mike Bearer because he was a clinical microbiologist. He had an interest in TB infection. David was then connected to Ian Hancock because they worked with Sir James Badley. And Sir James Badley at the time at Newcastle was quite instrumental in the 60s, 70s in terms of working on teichoic acids in Bacillus subtilis. And he wanted to work on TB at the time, Ian Hancock did. So David at the time sort of thought, we've got all these different individuals doing different things. And then he sort of knew that I was going to come back to the UK. So he engineered it. It's quite an interesting story. He was sitting in a lecture theatre in Newcastle in chemistry, and he was listening to a candidate who was interviewing for a chair in physical chemistry. He was sitting there and he thought, he goes, if we can recruit someone like this to physical chemistry, why can't we recruit someone like Dell to Newcastle? And he bumped into the Pro-Vice Chancellor for research in Newcastle at the time, and he mentioned it to him. And this is a true story. So basically, on a Sunday morning in the States, I get woken up by my wife saying, there's someone on the phone from the UK who would like to talk to you. And I says, well, it's probably my parents. Tell them I'll call them back later. It's obviously a lot cheaper to ring from the States to the UK than vice versa. And she goes, no, it's someone who's got a very, very posh accent and he wants to talk to you. I had no idea who it was. So I get up, go down, answer the phone, and turned out to be the Vice Chancellor from Newcastle University. He basically said that he'd heard from individuals that I was thinking of coming to the UK. When can I come and talk to him? So really, in terms of the collaborations and things, a lot of it's through word of mouth. A lot of it's through connections that you build up. I think one of the things I've learned over the years is that your PhD supervisor and the person whose lab you go into work afterwards as a postdoc, they can be hugely instrumental in terms of how your career shapes. So it's always good to sort of pick PhD supervisors are going to be very, very supportive. And also postdocs where you do your postdoc, where those supervisors are also going to be supportive. Handling a collaboration, those collaborations. I think in the early days, it was quite easy because there was a consortium of 5 of us together, and we could handle, individuals coming to us, talking about collaborations. And I think strength in numbers is always the key. Critical mass. I think when you have critical mass, collaborations tend to flourish. More people sort of gravitate to you because they feel that, you can actually take on board additional collaborations because you're a well-funded lab and so on. So I think critical mass was also key at the time.
Okay, thank you very much for that. That was really insightful.
I have a question.
Sure.
So I was wondering how often do you see patients having co-infection of HIV with MDR-TB, and what are some of the lines of treatment in such cases?
Yeah, so I won't say it's a huge number. And I can't say the number off the top of my head, but I don't think it's the major sort of influence in terms of MDR-TB. But obviously, I think when you have co-infection with HIV, I think then you have issues in terms of therapy, in terms of the toxicity of some of the drugs that are used, in particular the sort of injectable second-line agents.
Thank you. Another question. what are the challenges that a new researcher, when they land into their first job, say as a faculty, what are the things that they should consider as their top priority? There are a lot of distractions, but what they should focus on so that they can move ahead in their career?
I think I think one of the things to do is always think about, that balance in terms of when individuals start their careers. I was very fortunate in that, I've always had a fellowship or, a mechanism that's allowed and freed me up from teaching. So I think in a typical trajectory, when there's that sort of three-pronged sort of academic post in terms of you have research, teaching, and administration. I think in the early days, I think it's quite key to negotiate that your teaching and your administrative loads are fairly low, because then that will allow you to do a lot more research. I think also when you start out, I think the thing to do is have a focus. I think not to be too scattergun in terms of projects that you want to do. I think best example I can give is I had a postdoc work with me in the early days, and he had this mindset that basically he could almost see the research paper. He would actually think about the research project, think about the research paper, and actually start to assemble the figures on paper in terms of the experiments, in terms of what he wanted to do. So he was very much driven in terms of experiments that would then support figures for papers and to make a story in terms of research publication. I think that was really good because he was very successful doing that. And that provided him with the focus in terms of knowing exactly what he wanted to do. But it's tricky because you have to balance things, because you could be doing something that it may not work, it may give you very negative results. So at the same time, I always think that it's good to have two projects running simultaneously, one sort of more low risk, one more high risk. That's also beneficial as well.
Thank you so much. It was very insightful. Please feel free to unmute yourself and ask questions to Professor Bestra. We are very fortunate to have him today, so please feel free.
Everyone's very shy today.
Looks like it. Here it's around 4:20, around 4:30. So I think they may be a little bit tired as well.
Andy, what do you do at Newcastle?
Myself, I'm currently finishing my PhD in microbiology in the laboratory of Dr. Kevin Waldron. So metals in microbes.
Okay.
And we have now sent a review of metal in pathogenesis, and we cite some of your papers, and it's been accepted in PLOS Pathogens yesterday. So you get some citations from us.
I don't think it's because of my citations. I don't think so. No, that's excellent news.
But yeah.
So is that your first publication, or?
Oh no, it's my first author paper.
Great.
It's been a very productive PhD. So
that's great. So where were your other papers published, and what were they on?
Sorry, say it again.
What were your other publications?
We have one in mole micro, and then we have one in FEMS microbiology ecology.
Okay.
Again, talking about metals in bacteria and how it interact with pathogenesis and also survival in the environment. So we are kind of trying to strike both tunes. And yeah, we've done some reviewing of tuberculosis. We haven't done any experimental work on it, but we are trying to set up. So if you receive an email, you or one of your collaborators receive an email from Dr. Kevin Waldron, that's my supervisor.
Okay.
So yes.
Professor Besra, is there any possibility of having, say, international collaboration with projects that are, where the researchers are in line or that they want to work on a greater challenges around the world, like AMR and along those lines?
Yeah, so I think the Royal Society have sort of exchange mechanisms. I think they have mechanisms where they foster international collaborations, and they have exchanges where postdocs or students can come and visit laboratories in the UK. So that's one mechanism. I'm not sure whether the Wellcome Trust has that mechanism now. They're changing their funding schemes at the moment to discovery awards. I'm not sure that they're open to everyone, but I think there was something in there that, particularly from India, for instance, I don't think they're open to India because they feel that India has a very sound science base and has an infrastructure that's quite supportive. I think the Royal Society is the only one that I can think of off the top of my head where they have sort of funding mechanisms in place for international exchange.
Thank you very much for your answer. Any student want to ask any question? Please go ahead. Yes, I think the students are kind of shy today. I don't see any hands now. Is there any email that we can reach you, at a later point of time?
Yes. Would you like me to put it in the chat?
Yeah, that would be great.
Okay, there we go.
Okay. So kindly note Professor Bestra's email address. In case if you have any questions, please email him with your queries. I don't think we have a lot of questions today, but I really like to thank you for this wonderful talk that you presented here. We have a lot of participants who are either on YouTube or Facebook Live, so we may not have so much in the Zoom here. So that's so I like to thank the Microbiology Society for giving us this platform where we can share our ideas, our work, and also I like to thank my university, Adams University, for giving me this platform as well. And I like to thank all the participants today for participating, those who are either on Zoom and also they are on Facebook and YouTube as well. Finally, I'd like to thank once again Professor Bestra for his wonderful talk. Thank you so much, sir.
Thank you.
Thank you, everyone.
Okay, bye now.